So they have animal viruses, and as we've talked before, viruses only have DNA or RNA as their genetic material. So the general phase in the life cycle of both animal viruses is absorption, penetration, uncoating, synthesis, assembly, and then release from the host cell. And the difference in the two DNA versus RNA virus is the RNA virus first has to use reverse transcriptase. So they have to make DNA in order to use RNA. So the first phase is absorption. And so viruses, like we said earlier, they're obligate intracellular, so they need a host in order to reproduce. So they have specific host range due to their receptors, such as spikes. So the invasion begins when the virus encounters a susceptible host and absorbs specifically to the receptor sites on the cell membrane, those being the spikes. And a virus can invade its host cell only through making an exact fit with that specific host. So it's very specific in which it can invade. And it can be broad or restrictive host range just depending on what kind of virus it is.
So then we have penetration and uncoating of animal viruses. So we have endocytosis, which the entire virus is engulfed by the cell and enclosed in a vacuole or vesicle. Usually, animal viruses are, um, they have cellular membrane taken from animal host cells. And then uncoating, the enzymes in the vacuole dissolve in the envelope and capsid, and this releases the virus into the cytoplasm. So, with synthesis, replication, protein production, assembling of mature virions, um, the viral nucleic acid takes control over the host synthetic and metabolic, whatever that word is.
Okay, so now it's the release of mature viruses, right? Okay, yeah. So this is when the envelope viruses exit the cell by budding or exotosis. I don't know how to pronounce. Um, Viruses are shed gradually without destruction of the cell. And then damage to the host cell as a result of viral infection, diagnostic meaning. Um, Cytopathic effects that damage to host cells. Viruses induce damage to the cell that alters its microscopic appearance. Damaged cells look differently from healthy cells. Pap smear, that's the cancerous cells, no, clue cells infected with papilloma viruses that might cause cervical cancer to look different. Cells undergo big changes in shape slash size and acquire inclusions. And then inclusion bodies of viruses or damaged organelles and nucleus. Oh my God, I don't know how to pronounce it. And then syncytia, a fusion of multiple whole cells into single large cells with multiple nuclei.
Okay. Persistent infections. A career relationship develops in some cells. Cell harbors the virus and is not immediately lysed. Provirus is viral DNA, a comp wait, viral DNA incorporated into the DNA of the host. Um, Viruses and cancer. Oh, I'm definitely gonna mispronounce that word. Uh, I'm not even gonna try it. Experts estimate that up to 13% of human cancers are caused by viruses, and then transformation is cut off. Yeah. That's a learning outcome.
I have transformation in my notes if
Do
you want me to.
you?
Yeah, I
Yeah,
do.
you go through it. Yeah, because
Okay, transformation is the process of changing of normal cell to cancerous cell upon provirus incorporation into host DNA. So it goes with like oncogenic viruses, if that's how you say it. Do y'all want to do the concept check now? Oh, there is none.
Oh,
Can skip
okay.
it. Janaya, do you want to read one more slide?
Okay, yeah. Techniques in cultivating and identifying animal viruses. Viruses cannot be cultured using traditional bacteriology methods because they are obligated parasites, which means they need hosts. Special ways to be designed in vivo, which is live, and vitro tube, which is tube. In vivo methods, viral cultivation and lab animals are embryonic bird tissues for bacteria viruses, co-culture with live bacteria, in vitro, which is glass methods, viral cultivation in cell or tissue culture. Primary purposes of viral cultivation is isolate and identify viruses and clinical wow my specimens, prepare viruses for vaccines, do detailed research on viral structure, multiplication cycles, genetics, and effect on host cells.
Detecting viral growth in culture. There's degeneration and lysis of infected cells, which form plaques, and plaques are clear, well-defined patches in the cell sheet. They develop when viruses released from an infected cell radiate out to surrounding cells and infect those, and the infection spreads gradually and symmetrically from the original point of infection. New? Thank you. You can skip that one too. Viruses and human health. It is impossible to measure the number of viral infections in the world, and most common cause of acute infections that do not result in being hospitalized are the cold, chickenpox, flu, herpes, warts. Some, however, do have high mortality rates, such as rabies and Ebola, and other infections that lead to long-term debility, like polio and neonatal rubella.
Okay, so I think we're going to be talking about prions next. So prions are different than viruses because they have no nucleic acid, and so they have no DNA or RNA. They have a common feature of spongiform encephalopathias, which is distinct protein fibril forms in the brain tissue of affected animals. And a disease that prions cause is Creutzfeldt-Jakob disease, and this affects the control nervous system of humans, and it's a gradual degeneration of health and eventually leads to death. The transmission is unknown, but there are some similar diseases in animals. And the exact mode of infection is unknown as well, but the protein composition of prions has revolutionized ideas of what can make an infectious agent. And yes. And then we have satellite viruses, and they are dependent on other viruses for replication. So they need one virus to be present in order for their virus to be a thing. And one is called adeno-associated virus. It's thought that it could only replicate in cells infected with the adenovirus, but now we find that to infect cells, they have to be infected with other viruses or have had their DNA disrupted through other means. in virus or virus-like agent that parasitizes plants. It's about 1/10 size of an average virus, so way smaller, and it's composed of naked strands of RNA, so it has no capsid or any other type of coating, and this affects economically important plants such as tomatoes, potatoes, cucumbers, and citrus trees.
Okay, I think that we've hit our mark.
We have,
Awesome.
Is this a good closing point?
yes.
Okay, then I'll stop the recording here. Amazing.